So most of the rest of the so next I'm going to just go through a number of different examples of pathway analysis of different types of data, so you can get a sense of the interesting results that you can get from applying these types of analysis methods. So the first example is about autism spectrum disorder. So autism spectrum disorder has a important genetic component. It has over 60% concordance in identical twin studies, and at the time that this this project that I'm going to talk about was published a number of years ago in 2010. At the time, there were about 5 to 15% of the genetics can be explained by rare gene disorders. But at the time, people had kind of started to realize that a lot of the autism spectrum disorder phenotype could be explained by de novo copy number variants. So they'd started discovering these. And so Steve Scherer's lab at the Hospital for Sick Children, whose main focus is studying this autism spectrum disorder and the genetics of this disorder, decided to do a genome-wide, decided to look at de novo copy number variants to better explain the variants in the phenotype. And so they took about 1,000 cases and 1,000 controls, and they applied SNP arrays to genotype all the individuals. These SNP arrays measure the presence or absence of SNPs across the genome. And because his group was interested in copy number variants, what you can do with these SNP arrays is you can look for SNPs that are all, highly detected or not detected at all in a kind of string across the genome, and that can help identify copy number variants. So if you don't detect a SNP, a single SNP, well, that probably means that maybe there's another version of that SNP is present. But if you have a whole string of them, like hundreds of them that are all next to each other, like all the consecutive SNPs on the genome are all not detected, probably the explanation is that chunk of the genome is missing, and that's a copy number deletion. And everybody on Earth has copy number deletions and also amplifications. So those are detected by seeing the same kind of pattern in the opposite direction. So from SNP arrays, you can identify all these copy number or infer all of these copy number variants. And they were interested in the rare ones because those are more likely to be de novo and more likely to cause a serious phenotype. And so they took the ones that were present in less than 1% of the population. And there were some copy number variants or regions of the genome that were affected by copy number variants that were associated with autism spectrum disorder cases. But that only identified about 10 genes. So it wasn't really a lot of additional information compared to what they knew. So what we did was do a pathway analysis. So we took the copy number variants as a whole, and we asked if any pathways as a whole were affected by copy number variants. So let's say we take a pathway like cell proliferation, and we find that it has 100 genes, and we ask how many individuals have a copy number variant gain or deletion in one of the genes in that pathway. And if a single gene in that pathway was affected, we say, okay, this pathway is affected in this individual. And then we do that for all the individuals, and we count up the number of individuals of the cases that are affected and count up the number of individuals that are affected in that pathway in the controls, and we compare them with the statistical test that can tell us if there's a stronger association with the cases. There's more of this pathway more individuals affected having copy number variants that affect this pathway in the cases than the controls. And that actually painted a very rich picture of pathways that are affected or associated with autism spectrum disorder. So all these little circles represent pathways, and the color represents the association, how strongly enriched the pathway is, like, how different the numbers are between cases and controls. And mostly the pathways were affected by gene deletions or genome region deletions. And yeah, so all of these circles are kind of those pathways. The color is representing the strength of the association with cases. The size of the circle is the number of genes in the pathway. And these little labels are all the pathway names. And then there was a lot of pathways, so we grouped them into bubbles here and then labeled the bubbles, and that kind of reduced the complexity of all the labeling that we would have to do. We did label some kind of interesting ones that were just highlighted in the paper. So when we saw this, there was a lot of new information, and we didn't know if we wanted to kind of verify it. So one of the things we did was we looked at known autism spectrum related phenotypes, intellectual disability and autism. We collected genes that were associated with those phenotypes, like about 100 genes in total, and we did a pathway analysis on those gene lists. And all of these other like triangles are pathways that are enriched in the intellectual disability genes, and trapezoids are these other shapes that are represent pathways that are enriched in the known autism spectrum disorder genes. And what was interesting was that there was, even though there wasn't a lot of overlap between the known disease genes and the genes that were in the pathways that we found, they were frequently enriched in the same pathways. So intellectual disability affected central nervous system development, and autism, these copy number deletions also affected pathways that are involved in central nervous system development. So that helped kind of support the idea that all these new pathways were biologically relevant because they were somewhat related functionally with known disease genes. So another interesting, so you can kind of zoom in here and see some more details. One interesting thing to note is that when we looked at the genes that were involved in a given pathway, like cell motility, it wasn't the same gene deleted over and over again in multiple samples. It was different genes in the pathway deleted in different individuals. So, you might see that one individual has a gene, a cell motility gene, A deleted, and another individual has cell motility gene B deleted, and on and on. And eventually, by just kind of combining all that information with a pathway, within the pathway, you can get a strong signal that was not otherwise visible. So if you just looked at the individual genes, you wouldn't see this repeated effect between individuals. But if you looked at the pathway level, you did see this repeated effect between individuals. And that is kind of one of the key concepts, I think, of this workshop, is that by using prior knowledge like pathways, you can group information and make it stronger. So we sum up all of the individual counts that would otherwise be associated with an individual gene and wouldn't be significant. We sum them all up into a bigger number that ends up being significant. We'll talk about that more later. So feel free to interrupt with any questions that you have. I'm going to move on to the second example, which is about ependymoma. So ependymoma is a cancer of the central nervous system, the ependyma, which is the lining of the central nervous system. And this is a project that was led by Michael Taylor, who's a neurosurgeon at Hospital for Sick Children who studies this disease. Ependymoma in general is one of the more common brain tumors in children. Fortunately, brain tumors in children are not that common, but among brain tumors in children, this is one of the more common ones. And for a long time, people had known that it could occur in different anatomical regions in the brain. And based on where it occurred, they also knew that it had different outcomes. And in particular, if it occurred in the back of the brain, in the posterior fossa, which is the brainstem and the cerebellum, it was the worst possible anatomical location for outcome. And so Michael's lab studied this, and early on, just based on looking at gene expression analysis of all of these posterior fossa ependymoma tumors, had determined that there were two subtypes. subtype A, which affected the youngest individuals and has a terrible outcome, and cell type B, which affects the older individuals, still children, but had a actually great outcome. So they wanted to look at this more, and they did a whole bunch of genome sequencing and couldn't find any mutations associated with this disease. So, mostly when we think of cancer, we think that there's going to be a lot of genetic mutations involved because one of the hallmarks of cancer is genome instability. In pediatric tumors, it's actually not the same, especially early pediatric tumors. There's not a lot of mutations. So, it wasn't so surprising that we didn't find any mutations that were associated with the disease. But unfortunately, it didn't tell us anything more about the mechanisms involved. So we moved on to DNA methylation, and that was actually very informative. When we looked at DNA methylation, there was a very big difference between promoters that were methylated more in the A type, which is the serious subtype, and compared to the B type, which is the less serious subtype. So the interpretation was that in posterior fossa A, there were about 2,000 genes that were probably transcriptionally silenced by CpG island methylation. And when we looked at the genes involved, they didn't really tell us a lot. It was just 2,000 genes. It was hard to kind of identify what was common between them. So we did a pathway enrichment analysis, and we found that there was basically just one pathway that was really enriched. And it was, I'll tell you how we did this a little bit more, but it was targets of a PRC2 complex, which is the polycomb repressive complex 2, which is involved in methylating histones, and then DNA can be methylated in response. What this chart shows is different gene sets. So these are targets of the EED protein, so it's 12 protein, the PRC2 complex, and these two proteins are part of this complex. So all of these gene sets are all related. It turns out we're related to the PRC2 complex. The length of the bar here is proportional to the significance of how enriched the list of targets were in the 2,000 genes that we found to be differentially expressed. And everything that is bigger or above this green line was a kind of threshold of the significance that we chose. We considered significant, and there were only a few pathways significant in the A type, and there were no pathways significant in the B type. The way that we plotted these is that we converted a p-value. So we'll talk a lot about p-values in the workshop, but basically a low p-value means that there's is very unlikely for these targets to be enriched by chance. So that means that it's very significant. But to make it easier to visualize, we take the negative log 10 of the p-value, actually the multiple testing corrected p-value. We'll talk about that as well. And that converts the p-value into a positive number that is easier to plot on a bar plot. Just to summarize, the take-home message is that everything above this green line is significant, and there was only one real pathway that was coming up, which is this targets of PRC2 complex. And interestingly, before this study, there was no known mechanisms that were associated with causing ependymoma, and there's no treatment other than radiation and surgery. And so there's no targeted drugs or anything like that. So we were able to find that a bunch of chemical probes were available to inhibit the polycomb repressive 2 complex, and that killed posterior fossa A cells specifically in a couple of different models. And because there was no known treatment, it was easy to start a clinical trial. Nothing to compare to. So it turns out there's an on-the-market drug that targets the general process of DNA methylation, and it's called 5-azacitidine, and it's originally was developed for a blood disorder, but because it's safe in the clinic, the research team involved and the clinicians were able to get it used on compassionate grounds in a patient who had reached the end stage of their disease. So this was a patient at Sick Kids who is 9 years old, who his ependymoma had metastasized to the lung. That's this mass here, and in 2 months it doubled in volume. And then at that point there was no other treatment options available. Patient was in the hospital, not able to do much. So they applied this azacitidine, one course of treatment of this azacitidine drug, and that actually stopped the tumor growing for 15 months. And the patient was able to regain their energy and leave the hospital. And that was enough to kick off a couple of clinical trials that can test this more generally, also at an earlier stage. And those are still actually ongoing. They generally show promise. And so the nice take-home message about this is that we were able to, kind of the big picture take-home messages, we were able to go to knowing nothing about the mechanism of this disease to a drug that was actually had some interesting clinical effect in about 2 years. And there were a number of links in the chain to go from one to the other, from the raw data, all this data that we collected, to that. One of them was identifying the actual genomic data layers that have the information, the interesting information, but also this pathway analysis was an important link. If we didn't have any of those steps, including the pathway analysis, we wouldn't have made the connections that allowed us to identify the targets to study. Okay, so I am going to switch topics to, well, still talking about ependymoma, but a different type of pathway analysis. So in this study, with a few groups internationally who study ependymoma, they were interested in a molecular classification of all different ependymal tumors, including this posterior fossa one that I mentioned, but there's also others. So they collected data from a whole bunch of different types of tumors, and they clustered them and classified them into 9 different subtypes. All these colors here, there's nine subtypes here. Posterior fossa is the PF ones. And then we did a pathway analysis on each cluster, and then we summarized the whole thing as one of these enrichment maps that we'll learn how to build yourself in the workshop. But the nice thing about this map is it shows an overview of the entire disease in terms of which pathways are affected. So each circle is again a pathway, and we group them into bubbles with just to minimize the amount of text on the screen and make it easier to read. So these big labels are the main types of biological processes that are being affected, and you can just see the colors here that there's, the cyan type of tumor, it only affects, stem cell different, it's only enriched in certain pathways. And so there are certain pathways that are unique to specific subtypes. There's other pathways that are present across multiple subtypes, like these ones here. And so this just gives a really nice overview of the entire space of functional effects that are mechanisms that are likely important in these tumors. Okay, so I have a few more examples. The next one is about single-cell RNA-seq. So this was from a study that we were involved in to map the healthy human liver, and we had 5 liver samples. We had liver samples from 5 donors, and at the time, this was published in 2018. We were able to generate around over 8,000 single-cell RNA-seq measurements from all these samples, and this was the first healthy liver map that was available, and so we contributed it to the Human Cell Atlas project, which is a big project involving thousands of labs across the world to map all the cells in the human body at single cell resolution. So in these studies, all of this data can usually be grouped and clustered and represented as a 2D plot. Here, each dot is a cell, and the cells are grouped by similarity of their gene expression profiles. And normally what that does is groups cells of the similar type together. So you can see hepatocytes are here, and central venous endothelial cells are here, periportal endothelial cells are here, etc. So uh, and then this 2D map kind of just summarizes the relationship of all these cells to each other. So one of the things that's known in the liver is that there's an anatomical gradient as the blood flows through the liver. Blood in the liver kind of filters and cleans the blood, and nutrient and oxygen rich blood comes in one side, and actually, yeah. So there's gradients of nutrients and also oxygen that occur as the blood flows through the liver from it coming in, getting processed, and then going out. And endothelial, a number of different types of cells line this tubing basically that has these gradients, and a big chunk of those cells are hepatocytes, which are the workhorse of the liver, which have hundreds of functions that condition the blood and clean things up. So one of the things we wanted to do, we identified a number of different hepatocyte cell types or subtypes, and we wanted to see how they functioned across this gradient. So we organized the hepatocytes along a gradient that was known. So this gradient was studied in mouse, and people had found the gene expression profiles of hepatocytes along that gradient. And so we organized our hepatocytes along that gradient. So we can say we can have periportal on one side and pericentral on the other. And then we did a pathway analysis for each of these hepatocyte groups, and we summarized them again within an enrichment map, these little circle diagrams. And forgot to say one thing about enrichment maps, which is that the lines between these these circles represent overlap between the pathways, and that's actually what helps group similar pathways together. So basically, one of the main reasons why we make these is to kind of group similar pathways and reduce the complexity of the pathway enrichment analysis results. Here we can just see that there are different functions that are enriched in different hepatocytes. And so that conclusion for us was that the pathway analysis identifies a functional specialization of hepatocytes across this anatomical structure in the human body. Okay, so I'm gonna go through another example, but it's just a theoretical one, not based on a specific paper, but illustrating an important point that I've mentioned already, but just to see a little bit more how it works. Let's consider a genome-wide association study. So in genome-wide association studies, we collect a bunch of people, we split them into cases and controls. The cases have some phenotype of interest, the controls don't, and they're usually matched in some way, like age matched, and then we look at the mutations to see if there's a mutation that's associated with the controls, the cases, and not the controls. So here we have a bunch of SNPs, and say that we have 6 SNPs, A to F, and we have a bunch of cases and controls. We have 10 individuals, 5 cases, and 5 controls. And we look at the SNPs, and we say that there's a 1 in the column if that SNP A is present in the cases, and a 0 or in this individual here, and a 0 if the SNP is not present or not detected in that individual. And then we can look at these mutations and say, oh, this is really interesting. All the cases have SNP A, and none of the controls have SNP A. So that's a perfect association of the SNP A with the cases. Or we can see, vice versa, SNP D is present in all the controls and none of the cases. So that's, again, perfectly associated, but with the controls. So that's the ideal situation. It would be great if things were so easy, but actually most of the time we see something like this, which is that each case has a different mutation, and maybe we see basically every individual has a different mutation. So what do we do now? What can we do with this? Like, there is no mutation that's repeated. We can't say anything. Like, we've done all this work, and it looks like we have to throw the results out because there's no information. So any ideas?
Mutations might fall in the same pathway.
Yeah, so that's a good idea. Okay, so that's that's the answer. So if we organize the mutations, if we link the SNPs to genes and we organize the SNPs by if we organize those genes by pathways, we can collapse all of that information. So we collapse all of these ones here. Let's say we find that they're all part of the same pathway. We collapse them down into a pathway row. And so now we save the project because now we see a really strong association between our pathway. It turns out all those SNPs were involved in pathway apoptosis. And so we see that apoptosis was affected in all the cases and none of the controls. So, the way that works, so, we didn't just magically create information. The way that it works is that because we knew that those genes are part of that pathway, that's important information. It's prior knowledge. And that helps us, it can increase statistical power by two ways. So one is that we aggregate the counts. So instead of having one count per SNP, and we can't really do much with that, we now have 5 counts per pathway without doing any extra measurements. And so that aggregation of all those ones into a bigger number of 5 helps increase the power. And we also reduce multiple testing because instead of having to run tests with each SNP individually, we now run tests with just one pathway in this case. Of course, there'll be more pathways in the real analysis, but there'll generally be fewer pathways than SNPs. And we'll talk about that multiple testing a lot later. And the other interesting thing, so that increases statistical power via those two ideas. The other useful thing is that it generates a mechanistic hypothesis. We now can suggest that apoptosis is related to the case phenotype, whereas before we'd say SNP A is related to the case phenotype, and well, that doesn't tell me as much. I'd have to go do extra work to figure out what that means mechanistically. Okay, so here's another example, just quickly. Let's say we study gene expression differences between two conditions, and we find 1,000 genes that are differently expressed. What do they have in common? Well, we might find that they're all known targets of a transcription factor or a microRNA, and so if they would then be enriched in targets of that transcription factor, and then we might be able to say that transcription factor is explaining this pattern. It's upregulating these genes and activating these genes and repressing these genes, and so that might give us again a mechanistic hypothesis to work with.